Hello friends, welcome back to my channel Barufrig. Let's talk about C4 cycle and the Kranz anatomy of C4 plants. So basically what is the need of C4 cycle? Plants do have C3 cycle, right? But C3 cycle had a drawback. These plants were undergoing photorespiration. So C4 plants here have evolved in a way that they will just not perform this wasteful process. Instead, they basically undergo this particular C4 cycle. Now in the C4 cycle, what is the idea behind C4 cycle is to store more amount of carbon dioxide as compared to normal C3 plants. So in this way they can store a lot of amount of carbon dioxide and avoid the wasteful process of photosynthesis. Now in order to do so, they must be somewhat different. Their anatomy must be different from the C3 plants, right? So what is the normal anatomy of a C3 plant is that they have a epidermis. There will be stomata here, then again these epidermis cell, then they have palisade, mesophyll cells, then they have a vascular bundle, and they will have bundle sheath cells. Now remember, these bundle sheath cells do not have any chloroplast in them, so they cannot basically perform any photosynthesis here. In addition to this whole structure, they have spongy mesophyll cells, and those will have chloroplast in them, and they can perform photosynthesis. So overall, they just have spongy mesophyll cells and the palisade cells. This can also perform photosynthesis. Now, what is so special about the C4 plants? Now, C4 plants show something called as Kranz anatomy. Now this Kranz anatomy, basically these plants will have epidermis like this, okay, and they have a vascular bundle like this, okay. So they'll have a vascular bundle, and then bundle sheath cells which are there, these are bit modified ones here, and they do have chloroplast. Here they didn't have any chloroplast, right? They were not able to undergo photosynthesis, but this vascular bundle sheath cells here have chloroplast, and they can perform photosynthesis. In addition to this, they also have spongy mesophyll cells. Now the spongy mesophyll cells can also perform photosynthesis. Basically, this enables the plants to store more amount of carbon dioxide here as compared to this C3 plant. So this is a C4 plant, and it's showing Kranz anatomy. Now let's get started with the C4 cycle. Now in C4 cycle, what basically happens is that the atmospheric carbon dioxide here is taken inside the mesophyll cells. Okay, now we are dealing with the mesophyll cell and the bundle sheath cells. So there is a mesophyll cell there and bundle sheath cell. Now in the mesophyll cell, remember that there is no enzyme ribosco. Okay, and here as well there is no enzyme. Here there is, and here there is enzyme ribosco present. Okay. So here it is not there, and here it is there. Now in the mesophyll cell, as you can see, the ribosco is not present. In the bundle sheath cell, ribosco is present. So even if we take this carbon dioxide inside the cell, ribosco won't be there to act upon and convert your RUBP to the and divert that pathway to the Calvin cycle. Now this carbon dioxide will be taken inside, and carbonic anhydrase will convert it to the bicarbonate ion. Now this bicarbonate ion will be added to the phosphoenol pyruvate. Now this phosphoenol pyruvate that you see here is a 3-carbon molecule. When you add one more carbon to this, you get oxaloacetate, which is a 4-carbon compound. Now this oxaloacetate basically travels inside the chloroplast of the cell. Okay, although it's not shown here, just remember that this whole steps outside are happening inside the cytosol, while the oxaloacetate, once it's formed in the cytosol, will travel into the chloroplast. Inside the chloroplast, it will get converted to another 4-carbon molecule there, that is malate. Now this malate will now be transferred to the bundle sheath cells. Now this bundle sheath cell, this transfer is done via something called as plasmodesmata. Okay, now there is plasmodesmata in plants. I hope you guys are aware of this plasmodesmata. Now, once this malate enters bundle sheath cells, these are converted into the pyruvate. Okay, now the malic enzyme acts upon malate, and this is broken down into the pyruvate, which is again 3 carbon compound. So your malate was 4 carbon, it's getting converted to 3 carbon. Where has 1 carbon gone? It is released in the form of carbon dioxide. So that's how the plant is storing its carbon dioxide in the bundle sheath cells. Now here we are basically releasing 1 NADPH. Okay, now once this pyruvate is formed, it is again entering the mesophyll cell there. Once it enters the mesophyll cell, it's getting converted to phosphoenol pyruvate again. And here pyruvate phosphate dikinase, which is there, is breaking down 1 ATP there. Okay, and that's how you basically get back your phosphoenol pyruvate, and that's the C4 cycle. So the basic concept here of C4 cycle is enzyme ribisco is not present here in the mesophyll cell, it is present in the bundle sheath cell. That's how you can take in carbon dioxide and you can divert it in this path. Basically it can act upon the phosphoenol pyruvate now, and then as it enters the bundle sheath cell, okay, that particular extra carbon will be released in the form of carbon dioxide again, and it will be stored in the C4 plant, and it's completely avoiding the wasteful process of photosynthesis. So that was it, guys. I hope this concept is clear, and see you in the next lecture video.